me, that the really critical thing to get this all happening is really to get the controlled infection models going, because, then we can quickly test whether your kind of more, let's call it classical approach works. If so, great. If not, we're fine. The issue will obviously be, do we get it? Because I think if we do not make this happen, then we are in trouble. The last trial took forever, was incredibly costly. And I mean, you cannot always wait pretty much 5 or 7 years. Andrea can tell us how long it took from conception to conclusion, to know that, okay, this was not the right idea. Let's move on to the next. So that seems to be maybe one of the key things we should have in the discussion afterwards in the round table, because that completely changes our planning. If we could, and Bali said also, right, we should test quickly, and I completely agree. Even from failure we learn, but we can only learn if we can do them in different approaches and relatively quickly, in well-designed, obviously.
I agree.
So since no one else came forward, then maybe we can go straight into our discussion panel. Obviously, Mike will be a part of it. So Stephen will lead this discussion. Stephen Fong from Stanford, Chris Walker from Nationwide Children's will be part of the panel, David Thomas from Hopkins, and Marion Major from the FDA. But I think the other panelists also might chime in potentially. So Stephen.
Yeah, thank you. The terrific presentations for the past 2 days have highlighted really quite promising approaches for vaccine development. I believe that there is now a consensus that to succeed, vaccines should elicit broadly neutralizing antibodies and T-cell responses, recognizing the diversity of HCV isolates. We definitely need more powerful approaches to assess immunity that could provide new insights to determine the quality of vaccine responses in preclinical and clinical HCV vaccine studies. However, there are notable challenges in assessing HCV efficacy in humans. There are fundamental questions which are highlighted here that really we should address, which is really the focus of the panel discussion. The 3 questions are: how do we design a universal vaccine? How to recognize a potentially useful vaccine in the absence of a preclinical challenge model? And 3, when to advance an HCV vaccine to human trials. Next slide. In the first question, is it possible to design a universal vaccine? I like to call on the B cell side, I like to call on Manson, Justin, and Jans in that order to address the first question. Is it, can we design a single vaccine sequences that elicit neutralizing antibodies against the diversity of isolates among each and all genotypes? And clearly, Justin has presented very elegantly that we need to concern about serotype or phenotypes. And then the second question is, if the answer is no, what type of a multivalent vaccines would be needed? And Mike has really addressed that in his talk. So Manson?
Yeah, I think for science, everything's possible. I think we have to be optimistic about that. But I feel one thing important is every time we do some immunization, we need to find out what's the outcome and results. And also, I think Stephen and Justin, we all agree we have to standardize some of the assays so that we can generate compatible results and keep improving based on that. I think the field is going to a good direction in there. I think Justin developing this panel of virus. I'm piggyback developing the bunch of antigens that can be used in immunoassays and standardize the method so everyone uses the same method so that we can improve upon that. I think it's really the key. So I'm very optimistic. And in terms of whether a single sequence or mix and match type of approach, I think we've yet to see, but at least on my end, like noticing this so-called the multi-donor class of broadly neutralizing antibodies, to me is pretty groundbreaking in the HCV field. And we can try one of the major approaches in my lab is try to target this type of body neutralizing antibodies, see whether we can improve their yield and production, and also whether we can find a way to specifically elicit this body neutralizing antibody. So currently, of course, we are doing this single sequence approach, and maybe we need to mix and match, but those can be tested at least, like for example, in the monkey model. And so I'm very optimistic about it, yeah.
Mm-hmm. Justin?
Yeah, I agree with Manson, and I mean, I would say we don't know the answer whether a single strain or isolate is going to be adequate to induce broadly neutralizing antibodies. I really actually agree with Michael that it's the single, HCV1 or H77 isolate does surprisingly well at inducing cross-neutralizing antibodies. We do see that people who are repeatedly reinfected with different strains of virus do develop more broadly neutralizing antibodies than people who are infected chronically with a single strain. So I think that it's likely that a multi-isolate or mosaic antigen might ultimately be better. But I agree with Michael that we actually don't know, and it's still possible that a single isolate would be adequate.
Yeah, I think, thanks, Justin. Could I comment, Stephen?
Absolutely.
Yeah, thanks, Justin and Manson. Congratulations, your work is beautiful. Really enjoyed listening to you. It's really, given that it's E1E2 and it's a hell of a protein to make, and congratulations to Thomas first on getting some progress on the secreted antigen, E1E2. But it's relatively easy to make a combination of E1E2s from different genotypes. I think we know how to do that now. I agree. I think using your panel of 15 key pseudoparticles, let's try and show what is the best combination for neutralizing those. We have to bear in mind, of course, that if some are less well neutralized, does that mean it's going to be ineffective against those viruses? So maybe I would imagine, based on your talk this morning, that you will not be able to predict, perhaps, if a low response to some of your 15 pseudoparticles means they're not going to be effective. Is that correct? Would that be your interpretation?
Well, I mean, one thing I can say is that we've tested sera from a bunch of humans who spontaneously cleared HCV, and not a single serum sample neutralizes all 15 of those pseudoparticles. So I do think that, requiring a vaccine to neutralize all 15 is probably too high of a bar. I think the use of the panel is just so that we can compare apples to apples across different strategies, and whether it's 10 out of 15 neutralized or 12 out of 15 neutralized, at least we'll be able to see which vaccine seems most promising.
And if I may, Stephen, shut me up if I'm talking too much, but really would like to ask Justin and Manson, I presume you would want any envelope glycoprotein antigen to be used as a vaccine, to have these neutralizing antibody epitopes that require both E1 and E2. Do you agree that that is a criteria for advancing an envelope glycoprotein into clinical trials or not?
The short answer is yes, I agree. We've looked at the antibody types induced in people who clear versus persist, and we see AR4A-like antibodies in people who clear. So it does seem important to have antibodies that target this E1, E2 complex as opposed to E2 alone. I think it's still possible that if you got super potent antibodies targeting the CD81 binding site, which is just an E2, maybe that would be adequate. But I agree. I would advocate for E1, E2 as an antigen.
Yeah, I think your point really, we need to test out all this possibility. I mean, so far I feel the pipeline still relatively narrow, and also each time there's a candidate, I think finding out what sort of immune response is important. And we still do not know whether you need very diverse coarse neutralizing antibodies for protection or just one or two. But some work done previously in Charlie's and Alex, they showed that, of course, you have multiple body neutralizing antibodies will offer better protection than one. So if, I mean, a lot of the antibodies we found is to the E2 neutralizing phase, the receptor binding sites, and the AR4, which is at the interface, mostly on E2, and it's very broad. And that one, I think now when the structural information come, I think that's another area that I think should have a lot of intensive effort to see whether we can engineer that. Of course, naturally with the E1, E2 complex you make, and that produce very, very good, preserve the AR4 epitopes very well, I mean. And I think Thomas, maybe you can comment a bit on that, Thomas, first. Like, I think your earlier version of the soluble E1, E2, when with the LC dimerization domain, the AR4 was there, but wasn't as great as later on you improve further with the SC dimerization domain. So their way or their to further improvement, and once the thing's been improved, you really can increase and see the binding of AR4 much better, which is something that we notice, like when we make E1, E2, and then quite often the AR4 doesn't bind very well. It's indicative. It's a mixture of folded versus not natively folded proteins on there. Yeah.
Mm-hmm. Thank you.
Before Jan's comment, I'd like to raise a different point. Perhaps one of the difficulties of inducing BNAbs is that we know for some of these, at least, they are rather cryptic. Probably the best example was actually one of the first BNAbs that have been characterized by Arvind Patel and Jonathan Ball and their colleagues, and that is the AP33 epitope, or the AS412, which is the newer designation. And that we know that based on their work, they have shown that this sequence just downstream of H3L1 is highly, highly conserved among all isolates, not just within a particular genotype, but really among all isolates. But yet at the same time, only a tiny fraction of individuals that are naturally infected actually produce antibodies to that sequence. So the implication of that is that these that BNAb epitopes can be cryptic. And more recently, Jans have really described some really very important work. And I would ask Jans to comment in terms of, given the data that's out there, the findings that are there, what are, is it possible for us to stabilize or to better expose some of these BNAb epitopes in such a way that we actually can get higher affinity or broader breadth of these types of antibodies?
Thanks, Stephen. I actually don't know if I'm the right one to answer that, but I would just say how fantastic it is to feel this excitement there is for a vaccine. Just 4 years ago, many of us felt the necessity to get together to write a consensus paper on trying to convince everybody that it was important to develop a vaccine against HCV. So this development is really great. Also, as being the one that originally were the first to publish the 6 major genotypes of HCV, I'm of course a big proponent of the importance of these genotypes, both for diagnostics, for treatment outcome, and also for vaccine development. But still, at this point, I'm a little bit in doubt. The question was original question was whether we can get away with just one strain to vaccinate with, like Michael has done with the HCV1, and seen actually quite broad protection. Judith Godwine and her group in our COHEP program recently published a paper on a whole virus inactivated vaccine with just one strain. And there she saw excellent cross neutralization against, in that case, we tested against 7 different genotypes. And so that shows that just one antigen can at least give that kind. And I think that is also what we've seen in chronically infected patients early on. So there is that kind of cross neutralizing antibodies against the different genotypes. And I agree, neutralization is not determined by specific genotypes. It can very well, 2 different strains, closely related, can have very different neutralization profiles. So that's one observation. The other observation, of course, is that there is these things, as we also talked about yesterday, that I think Yannick Prantu also in our program several years ago showed that if you take the HVR1 out and then look at how these different strains are neutralized, the big difference between them completely disappears. So if the HVR1 sequence is gone, they all neutralize the same. So that kind of means there must be some epitopes in those sequences that are highly conserved. So if we could get at those in some ways, then that would certainly be helpful. And then finally, I think when it comes, I guess this was just with the antibodies, but I did want to make the comment also with the T cells. I think it was very apparent from early chimp studies, including one that I did myself, that if you have an animal that's infected with one strain, yes, you get, and we saw data also from Robert Lanford and Stuart Ray yesterday, if you're infected with one strain, you can get infected short-lived with other strains, but you many times will clear. And we did that with different genotypes also, and so did others. And what we found was that those animals had absolutely no neutralizing antibodies, yet they cleared one infection after the other, showing that the T cells alone could clear those infections again and again. So if we can combine those 2 components, the broadly neutralizing antibodies and those effective T cells, I agree with Michael that this is possible. Then coming back to your real question there, Stephen, I really at this point I'm not sure how we can. I think there's so much great work going on now on how to modify these envelope proteins, and I just think we need to see where it's all taking us and be very open to testing. And I really agree with that. We should not put any limitations. We should not say that because designer-based, that's wonderful, we can change the antigens, but I also agree with Michael, we should go ahead and test the authentic antigens in all kinds of formats. And then finally, I would just caution that if we have these, and I think it's great that these panels have been developed, both the CC panel from Pietzman's lab and the PP panel from Justin Bailey's lab, and they're really great tools to have. They're based on a lot of work from before. And I think what we just all have to remember is how important that when we use them, that we make sure, and that's especially true for the CC, that the envelopes don't change, because just one nucleotide change can give a 3-log difference in neutralization. So it's very important that we have the viruses that we use for neutralization assays under control.
Continuing on the T cell side, Chris, Arash, Andrea, Giora, would you like to comment?
It's Chris here, Stephen. Sure, thanks. I think that for T cells, we also don't know if a single sequence will suffice. As Jens just pointed out, a single infection with one genotype of virus can provide apparent protection from persistence with a different genotype. Whether we can achieve that with vaccinations, not known. I think it depends on how you approach the problem. There are clearly some approaches that involve identification of conserved sequences or epitopes across genotypes, and that those can be used. And I know that Mike Houghton and his colleagues at the University of Alberta and Ellie Barnes and her colleagues at Oxford are looking at those types of approaches. The other involve the use of consensus sequences, ancestral sequences, as championed by Stuart Ray for quite some time, and now epigraph or mosaic sequences from the Los Alamos group and our colleagues. It seems that at least with the mosaic type approach, a polyvalent vaccine will probably be required. It may be that will be easier to achieve with something like an RNA vaccine, which appears to give good T cell immunity, than it would be with construction of different viral vectors, although that's arguable. So a lot will depend on the approach, the vector system, how easy it is to make polyvalent. The one thing I will indicate, and I noticed there was a question in chat, it will be necessary to use the non-structural sequences as they are the dominant targets of the T cell response in HCV. So I guess that would be my comment.
Arash.
Yeah, thank you, Stephen. I think I'll follow up with Chris's point, and I guess, one important thing for me is, and I think Chris really is working on this topic as well, because having access to the actual site of viral replication in the liver these days is very difficult. So for me, the question is actually how many epitopes are being targeted by T cells? Is it, few at one, few, or many? And then how many are really required in cases in which either in humans or animals have resolved versus gone persistent? So I think for me, I still the question of whether we need to target multiple epitopes, although that what Chris beautifully showed that years ago, and that at least in the ones that resolved, they seem to be targeting multiple epitopes, but in actuality, in the liver, in the site of active viral replication, how many epitopes are being targeted and expanded in the liver? I think is a question that is of interest to me.
Andrea?
So I actually think that there is some evidence from COVID vaccination that's actually helpful here. I actually would say that the mRNA vaccines, as I think everyone knows, the platform was not initiated at the onset of the COVID pandemic. And in fact, Justin has been pursuing that in combination with the team at Penn, Drew Weissman and others, for HCV vaccines as well. But one thing that seemed apparent from the influenza vaccines generated with mRNA is that they don't induce CD8 T cell responses as robustly as vectored vaccines, for example. So I'm not sure that that is going to be the ideal platform for induction of CD8s. The other thing that I think is informative from the platform from the COVID-19 vaccine efforts is a recent study by Bob Cedar demonstrating that the ancestral sequence, which was really, the parent vaccine sequence essentially because it was made early in the pandemic, actually generated more cross-reactive neutralizing antibody responses against Omicron than an Omicron-specific vaccine did. And so I think that in the case of very diverse viruses, potentially from an antibody and a T cell standpoint, there is an advantage to using ancestral or consensus sequence. And that may actually be better than conserved epitopes, because some of these epitopes it is at least possible, as Chris, Georg, Barbara Rehrman, and others have shown, it is possible to escape T cell responses, including very commonly recognized T cell epitopes. So I think it's complex, but I do think we should learn from what's going on in development of other vaccines, influenza, SARS-CoV-2 vaccines, to try and inform some decisions. And I also agree with Georg. I think the one thing I will point out about the use of CHIM, because I will say, having conducted the first efficacy trial for a prophylactic HCV vaccine, it really led to people becoming very discouraged. And I do think that every failed vaccine trial has the potential to worsen that. So I want to be thoughtful about bringing vaccines into at-risk populations or we think we may really actually provide benefit.
Kira, any comments, additional comments?
I want to add a little bit in a different direction from what was said up, because I think the real problem for the T cells here in HCV is that just like in HIV, we have a really challenging virus, and we do not know what protective immunity on the T cell side is. Like, I don't know, and maybe same for the antibodies. We really don't know. It could be, we think good breadth and good magnitude is important, but it seems certainly not sufficient. I mean, Andrea's studies in her cohort and some of ours show we have patients with incredible T cell responses that become chronic. So quality is maybe just as important, or characteristics of, and what the vaccine can induce. And since we don't have the target of what we need, it makes it really problematic. So that's why I think Jim is so important, because the more things we can try, and hopefully the more we can learn, even if it doesn't work without having spent many, many years. And the other thing is, obviously, we always talk about CD8 T cells, and in HCV, it's pretty clear that without CD4 T cells, we have a problem for both the antibodies, most likely, and certainly for the T cells. And for them, our knowledge is on one hand even more clear, and that is without CD4 help that's sustained, there's no clearance, but at the same time, we know much less about these cells. So I think the best we can do is hopefully get these controlled infection scenarios started, plan them so well that we really get as much information about immunology out as we can, and then make better informed decisions if it doesn't work as we think it hopefully does.
Thank you. I think we should move on to the remaining questions on this portion anyway, and probably tackle both question 2 and 3 at the same time. And that is whether there's a common definition of a broad response, whether it's on the B cell side and the T cell side. Justin, if you could provide some additional comments, because you've shown in terms of like the importance of the diversity of panels in genotype 1, do you think that we need to do more for the other genotypes?
Yeah. I mean, I'm like a dog with a bone on this question. I think there is no standard definition of a broadly neutralizing antibody in our field, and that is a problem, full stop. And I think that we should work together, all of us, to define a broadly neutralizing antibody, either in serum or a monoclonal, based upon a standardized panel. And a lot of us already work together, Steve and Manson, lots of others, to try to come up with this panel that I talked about today. So I hope that can move us in the right direction in coming up with a standardized definition of a broadly neutralizing antibody. And I don't know the answer to the T cell, of course. I would defer to someone else on that question.
On the T cell side, who would like to take the lead on that? A common definition? Chris?
I think on the T cell side, the question of what a protective response looks like is still up in the air, especially on the CD4 side. Tobias Beutler and others have showed that follicular helpers, T helper cells, are an important component of the response. In immunogenicity studies, we tend to still look at Th1 type cytokines. It really seems like we may need to broaden the analysis, perhaps using the systems biology approach that Bally described. But I think, as Georg pointed out, the quality of the response is important. And as with antibodies, we don't have a standard definition of quality at this point, and what readouts we should be using. And I think it would be important to come to agreement on that.
Yeah, it looks like we're really running short of time. So what I'd like to do is move on to the next slide. Lindsay? Right. The next set of questions are really how to recognize a potentially useful vaccine in the absence of a preclinical challenge model. Bali has really elegantly talked about the lessons learned from the SARS-CoV-2. David, Mike, could you provide additional comments in terms of what you feel are the lessons learned and what more we could glean from those studies?
Was that directed to me, Steve?
Yeah, you and also David Thomas.
Oh, okay. Yeah, I'm not sure I can give a good response to these questions. I mean, we've already discussed, you know, what is a successful immune response at the B and T cell levels. And, you know, I think all we can do is kind of be practical, right? If we're looking at different vaccines, we would like them to neutralize many of these nice panels that Justin and Thomas Peachman have put together, right? If we have one that is very poor at neutralizing most of those, then we wouldn't be too enthusiastic about putting that forward. And the T cell, I must admit, I talked to some GlaxoSmithKline vaccine people a few years ago, and I told them the chimp data we had with E1, E2, and they seemed to be impressed. And I told them, well, we're also going to dial in T cell responses. And they said, well, why would you do that? Why don't you just go with E1, E2? And, you know, obviously at the time I was influenced very much by the great T cell immunology that's been done in the field. And I thought, no, we got to have T cell responses. And I still feel that, but I guess I am a little bit, I'm a little bit perturbed about the Icarus trial not working. And I guess I've been influenced by the studies I briefly mentioned in my talk this morning, that when we vaccinated 5 animals with our T-cell vaccine, a big polyprotein, very similar to what Icarus expressed in from their vectors, we expressed a big protein in yeast, you know, 2,000 amino acids. We combine it with a really good Isca-matrix adjuvant. Using Chris's great methods, we show that prior to challenge of those animals, we had multifunctional CD4 positive T cells specific for the virus in the liver. And we had multifunctional CD8 positive T cells in the liver prior to challenge. And yet when we challenged, everything went well for the first few months. Very low viremia, very low acute hepatitis. But boy, when we followed up those 5 animals, it took almost 2 years for one of them to resolve the viremia. The other 4 were persistently infected. And so I guess I'm just kind of a stream of consciousness here, but I do sometimes, I guess I feel it's very important, as Andrea's doing, trying to find out why that trial did not work. And I've been influenced by your work, Chris, that we know that CD8 responses, that the virus can evade those. And, you know, I think the quicker we find out if that's what was going on, then it's really going to help our field. And yeah, I'm continuing to dial in T cell responses with my colleagues. We think, I'm kind of assuming that the reason the Icarus trial failed is because the viruses evaded the CD8 response. And that is why our strategy is to really focus on small, highly conserved epitopes, T cell epitopes, and use really good adjuvants to prime those in people. And as I think I mentioned, there's a nice paper with TB antigens using AS01 that did prime very good CD4s, very good CD8s in humans. So I'm not saying we're going ahead with AS01 because there's some other issues with that it's a very reactogenic adjuvant. It's used in the shingles vaccine. A lot of people get quite sick from that shingles shot, and it's only 2 shots. Well, I believe, I guess I've been influenced by Hep B. I believe we need 3 shots for Hep C, and I'm not so sure especially people who inject drugs, whether they're going to come back to get all 3 shots if we use AS01. So whether we should use the one that the adjuvant that Bali's been showing nice data for, the Toll-7, Toll-8 agonist, or whether we should use the Toll-4 agonist needs to be decided.
Can I maybe answer to Mike quickly? Because I think even, for example, if we see now that there are various CAPE mutations in the trial, we need to be very careful. We need to find out, was it consequence of persistence or the cause, or was it the consequence of having T cells that did not have the quality, or did we not have enough CD4 help? Or so there are just a lot of opportunities. It is complex, and that's why I really hope that we can see a number of different controlled exposure trials in the next few years, that we can sort this out a bit, because there's nothing better than, mouse data and stuff are not helping us, obviously. We need to see what certain things do and what happens to certain immune responses when a human gets exposed. And if we can observe it in real time, because that's the other thing, obviously, compared to the regular trial, apart from being much less costly and shorter, et cetera, is that we can observe when the inoculum goes in real time what happens. We can do this work that Bali has pioneered, these kind of things. We can look at everything and know the sequence of events. And I think then we might be finally able to learn, what is needed and where exactly does the defense get cracks. And I think that will really be very informative. And so I just think it's too early to really, go on just put on the money on one horse.
Mm-hmm. Yeah, good points.
David, Bali, do you have additional comments for the few minutes that we have for this set of questions?
No, these are excellent comments, and I very much agree. And sort of highlighted how I thought what the lessons were at the end of my talk. So
I think we need to move on to the last set of questions. And that is when to advance a HCV vaccine to human trials. And these are the questions: whether requirements for vaccines to enter phase 1. And second question, what population should be included in the phase 1? Should we go beyond people who inject drugs? And if we think that would be the case, what would be that population? And lastly, what are the metrics to advance an effective vaccine candidate? Marian, would you like to take the lead on these questions?
Yeah, sure. Thanks. So the first question, and I think that there have been a lot of really important comments about moving into human phase 1 trials. And I really like the last point that Bali made in his presentation of basically looking at them as like phase 0. And all of these discussions about the human challenge model, I think they're important, but that's a massive amount of discussion that still needs to go on. But you don't have to wait for something like that to move something into a phase 1. And as so many people have said, and I think Mike has said, you don't know what's going to happen in terms of human immune responses unless you immunize humans. There isn't really a model that we can use to predict. And you don't need a huge amount of information in terms of what the FDA might require in order to go into a phase 1. We would want to see some reassurance of safety and immunogenicity, but a phase 1 trial is to test safety and immunogenicity in humans. We're not asking to be convinced that this is going to be protective. We're not asking that this is going to be moved into a phase 3 trial. That's for vaccine developers to decide if this is what they want to do. And so these things, it doesn't have to be sequential. And I feel as if sometimes we're getting a little bit too sequential with this. We want to sort out this problem before we move to the next problem. And there's so much information that can be gained, and you don't have to have the perfect candidate for a phase 1. and it's not uncommon for sponsors to come in with different candidates or variants of one candidate to see which one they want to take in to the next phase. So I really would like to see people thinking much more about what to take into a phase 1 trial. And finally, just to encourage people to come forward with pre-INDs. I don't know if people are familiar with pre-IND meetings, which would come before an IND application, but you can ask FDA a lot of these questions that you may not be sure of. What do you need? What do you need about immune responses? And the last point on that last slide about standardized assays, which is important, but again, they don't have to be established before you go into a phase 1. So I really think I'd encourage people to think much more openly about trying vaccines in humans at this point. Thanks.
Andrea, David, would you like to comment on this topic?
Dave, you want to go first and then I'll comment?
No, go ahead, Andrea. I think it's you did a study. I think you have the right to speak.
Well, and I think we all have a lot of collective experience. I agree completely with what Marion said. I mean, I think part of, and what Michael was saying, that I think, you know, it would be helpful to do head-to-head comparisons of things we've theorized might be advantageous. And we're trying to do some of this work in animal models, but we know the inherent flaws to that. So I do think that a head-to-head comparison of various strategies for inducing neutralizing antibodies, I completely agree with the need for consensus on what is defined as a broadly neutralizing antibody response. To me, I think you need a good neutralizing antibody response. I think you need T-cell responses as well. I don't think anyone thinks that there's going to be a neutralizing antibody response that neutralizes every variant of HCV to which someone might be exposed. So I think having a T-cell response to complement that is critical. I wouldn't make too much of the Okyris vaccine. It was a BK strain vaccine. Every circulating strain of HCV carries a number of escape mutations. That's how it circulates in human population. So I think there are numerous reasons that I don't want to extrapolate too broadly beyond the vaccine as prevention trial to say that a vaccine is not possible. I firmly believe it is possible to get protective immunity. And I think in phase 1 clinical trials, we could determine what our best in class T cell and B cell antigens are and bring that forward.
That was a lot more thoughtful than what I could have said, but I think just to synthesize the answer to your question that you just answered and the one that was asked before is, you know, what we learned with SARS-CoV-2 is that if you're in the middle of a public health crisis, that you get the smartest people you can to think about the problem, and then you act decisively, and you make sure resources aren't the limiting step. And then sometimes you're surprised by how well things work out. And I think that's what we learned with SARS-CoV-2. And I think we have a public health crisis right now. There's, instead of 6,000 people that are supposed to be acutely infected with Hep C in 2030, we're going to probably have more like 90 or 100,000. And we're not going to eliminate. So we need to respond and get the smartest people, think as hard as we can, and then, as Marion said, take a lot of attempts into phase 1 and see what we learn. I mean, that's my opinion.
I maybe want to add, because I think, Marion, your message is so important to be heard by more people, that it does not take as much as many people think to do a phase 1 trial. A lot of, I see this still so much. It's like, you know, therapeutic HPV vaccine is the same. People seem to want to modify and test their vaccines for years in mice and whatnot of models they have. And it frustrates me immensely, because we all know that's not going to help us. We need to see what quality of immune response we get in humans, that we have a baseline, also that we can compare different vaccine approaches and see what kind of immune responses they make, so we can make a better decision.
Yeah, I do want to make that point that a phase 1, there are many vaccines that do not go beyond a phase 1. Many more that do not go beyond phase 1 than actually do. And you can learn so much from them, I think, that can inform. So I guess one thing would be, don't be afraid to fail. That doesn't mean that you can't learn from it.
Well, on that note, let me ask what are the expectations and what are the metrics to advance beyond phase 1? To really people who have already made comments, and I mean, what is it that we're looking for, and in what population? Who would like to tackle that?
Well, I guess I'll have a go, Stephen, and you probably can predict my answer, and that is, if we can see in phase 1 pretty good cross-neutralizing antibodies to these panels from Justin and Thomas Peachman, that would be a thumbs up, right? And I'd like to see strong CD4s, obviously, but I'd like to see strong CD8 responses to highly conserved epitopes. And if, the more the better. If we've really primed CD8s to highly conserved epitopes amongst the genus, and we've got more than one, I think I would be extremely pleased and enthusiastic to take that into a CHIM or some other efficacy trial. Could I, if you don't mind, could I just come back a little bit? Because I would like to ask Andrea and Marion, your question on question 2. You're asking, should the phase 1 be in, I think you're asking, should it be in healthy volunteers or should it be in PWIDs? I'd really like to hear the comment from Marion and Andrea on that. The first phase 1 trial I'm talking about.
Well, I would comment there that a phase 1, the first phase 1, I don't think that PWIDs would be treated as different or special. I mean, I think that there's no reason to think that they're not necessarily a different population. They're not necessarily unhealthy. I'm not sure that that's the case. Andrea could comment on how they'd be classed. But one thing would be, if you are going to do that, and I think that might be difficult for the initial phase 1, just in terms of planning a clinical trial in PWIDs may be more difficult than in healthy population, but also it might be that because they are a vulnerable population, it may be considered that initially it should go into a healthy population. So I think that question, was more, I was thinking, well, maybe the follow-up, you might look at your phase 1 in a healthy population, in a non-PWID population, but then maybe a follow-up would be in that group to see if the immune responses are similar. That's my thought. Maybe Andrea can add some comments.
Andrea, quick comment.
Quick comment. I agree with that. I think I would do head-to-head comparisons of vaccine strategies in healthy volunteers. Despite the extensive expertise of Kim Page, Margaret Heller, people around the world, Nagala Shukri with Julie Bruneau, and following PWIDs, it is more challenging than following other populations. So it's easier to do a head-to-head of strategies in phase 1. And ours is one of many studies now with HIV vaccine trials showing lower responses in PWID. So I think you get your best response in healthy volunteers not at risk for HCV and move to PWIDs next. But one could argue that you may get more information by going into PWIDs first. It's just harder to achieve good follow-up and other things than in healthy control populations.
I'd like to close up this discussion to thank everyone and all the participants, all the speakers, and also for this very lively discussion. I don't know whether there's enough time for the organizers to make some closing comments. John, Chris, Rajan?
I think John has